In 2006, we chose Jackson Hole for a very interesting endeavor. I'd spent the last 30 years or so living in small villages, working on drug discovery programs. Fortunately, I was blessed with success. We discovered a new AIDS drug, drugs for inflammation, immunostimulation. But I wanted to go for a very specific disease called ALS. I'm just interested, how many of you have had a friend or acquaintance here that's suffered from ALS or Lou Gehrig's disease? Good heavens, that's a large percentage of the audience. The pharmaceutical industry, and I have a lot of respect, I've worked with most of the major players, but they take too long. It costs right now about $800 million to $1 billion to get a new drug on market, and it takes 10 to 14 years. ALS patients don't have that sort of time. Once you're diagnosed, on average, you have 2 years to death. So we decided to create a virtual pharmaceutical firm. I cherry-picked the top scientists. We got 27 universities around the world, from Tokyo to Stockholm, to work with us. We set up a first-rate laboratory. I like to tell people we have the best amino acid laboratory within 2 blocks of the Jackson Town Square. And I want to tell you a little bit about our success and what we've been doing. I want to start you off at Grand Prismatic Spring in Yellowstone National Park, one of my favorite places. What's interesting, although the Yellowstone Basin is geologically recent, those residents in that cause the colors are very ancient. These are cyanobacteria, photosynthetic bacteria. In this case, the yellow pigments on the screen are caused by the genus Formidium and Sinococcus. Now, these are probably more familiar to you as mats or scum that you see on low reservoirs or Gulf hole water traps. We have on the top from our laboratory a light microscopy photo of cyanobacteria, fluorescent photo. They're fascinating organisms. In fact, it's believed they're the oldest organisms on planet Earth. We have fossil record going back 3.5 billion years of continuous occupancy of cyanobacteria on planet Earth. Here they create giant blooms. This is a boat in the Baltic Ocean going through a mass population of cyanobacteria. Here's a satellite photograph of the entire Baltic. You can see that some of these blooms measure thousands of kilometers. So let me tell you a few quick things to remember about cyanobacteria. First of all, they're the oldest organisms on planet Earth. This means that they created the oxygen atmosphere of the Earth. Prior to the advent of these photosynthetic organisms, this would have been a lethal environment because of ultraviolet radiation. They create and continue to be the major sink for carbon on this planet. 2 nanoplankton genera, so small you have to have a microscope to see them, have the greatest capacity for absorbing carbon dioxide in the world's oceans than all of the rainforests combined. They are found everywhere, from the hot pots of Yellowstone to the Antarctic to the Gulf regions of the Persian Gulf. They're everywhere. And what we're very interested here at our institute is that they produce toxins. This is the toxin beta-methylamino L-alanine, or BMAA. The right part of the structure is the common amino acid alanine. But when cyanobacteria produce it, they add an extra nitrogen group and a methyl group onto it, thereby making it neurotoxic. We've discovered in our research that almost all cyanobacteria around the world produce this toxin. So, we also found that they live inside the roots of the cycad trees, and so I want to take you from Jackson Hole to the middle of the Pacific Ocean, where we've been doing work in the islands of Guam. Here's a root of the tree that I've cut in with my Swiss Army knife. You can see that green photosynthetic layer are cyanobacteria. We brought these back to our laboratory, cultured them, and found that they produce this neurotoxin, BMAA. So what, you might ask? Well, we got interested because we found that these are produced in these roots that are aerial, so they can get photosynthesis, and the indigenous people of Guam, the Chamorro people, have figured out how to take the seeds of the tree, take the kernel of the tree, or the gametophyte, and make flour out of it. We discovered that this flour is rich in the neurotoxin. And we started wondering if this could account for the devastating form of ALS that this village has. We've made mobile expeditions to the village. 25% of the people are dying of this disease. To put that in perspective, 1 out of 350 Americans dies of ALS. Imagine a village where a quarter of every adult death is attributable to this. What's even more interesting is that these people, like this individual on the screen, some have typical ALS symptoms, others have symptoms of Alzheimer's with cognitive loss, others show Parkinson's tremors, some poor individuals have all three formative symptomologies all at once. Could this neurotoxin be linked to this disease? We also discovered that large bats called flying foxes eat the cycad seeds and accumulate the toxin in their tissues at 10,000-fold the time that the bacteria produce. So when the people eat these bats, and they love to eat them, they eat the head, the wings, the fur. They love them so much that they extincted one species and put the other on the endangered species list. The only remaining bats are on Anderson Air Force Base, a nuclear weapons base. I've recorded 2 interviews with Chamorros who have taken their guns, gone under the B-1 bomber, being around the nuclear weapons bunker, shot a bat, and repeated the process to get out of there, suggesting to my mind that either these people have an insatiable appetite for flying foxes or that our much vaunted Homeland Security is still not up to snuff. So the hypothesis that we developed is that these bacteria, cyanobacteria, produce this toxin, BMAA. It accumulates in the trees, gets into the people, and kills them. But since cyanobacteria occur everywhere, could that be what's happening to ALS patients here in the mainland? Our colleagues at Dartmouth University are in a consortium. Dr. Elijah Stommel in the neurology department there started mapping ALS patient residences in New England. And what they found was totally stunning to me, that the ALS patient residents in New England tend to cluster around lakes and streams that are polluted and have cyanobacterial blooms, but not around pristine waters. In fact, the Dartmouth team estimates that if you live next to one of these streams, your risk of getting ALS goes up 25-fold. Now, what's interesting about ALS, Alzheimer's, and Parkinson's is that they're tangle diseases. They're caused by misfolded and tangled proteins. Could BMAA play a role in tangling these proteins? When I lived here, my dad was a ranger in the park. This was Christmas. I got a Slinky. Turns out a Slinky is actually a very accurate model of a neuroprotein. 20 different amino acids in different sequences are linked. Unfortunately, this is typically what would happen to my Slinky in Moose, Wyoming, after only about 2 weeks. Could BMAA cause a perfectly formed neuroprotein to generate a tangle characteristic of these diseases? Let me show you what happens. DNA has the instructions to assemble neuroproteins. Here we have serine coming in to the ribosome. Each of the 20 amino acids has a different tRNA that brings it in. What we discovered with our colleagues, Ken Rogers and Rachel Dunlop at Sydney Technical University, is that if BMA is present, it actually grabs the tRNA for serine and knocks it off and kinks the Slinky and causes human proteins to misfold. When we announced this discovery last year at the International ALS Congress, you could have heard a pin drop. Because we think, just by chance, we've stumbled on a major mechanism for protein misfolding. So our hypothesis is this. You're exposed to BMAA, it crosses the blood-brain barrier, it mischarges this tRNA, it gets incorporated in the protein, causes the protein to misfold. This causes the proteins to get sticky and aggregate. Meanwhile, it directly is toxic to motor neurons and causes ALS. But even more important was a discovery that just took our breath away. We discovered with our Australian colleagues that if we add excess serine to human cell cultures, that we can stop protein misfolding, we can stop the tangles, and we can stop misinsertion of BMAA. We discovered that the dietary amino acid L-serine blocks BMAA from inserting into proteins. Is it possible that a common dietary substance could prevent or possibly even treat ALS? We are now moving as fast as we can on this because, again, the patients don't have time. And so we are beginning this year trials of L-serine for 20 ALS patients. Our previous drug is already in phase 2 FDA-approved trials. This will be in a phase 1 FDA-approved trials. There's nothing I'd be happier with than to have a new treatment for this serious disease walk out of this town. I want to thank my colleagues who I'm so honored to work with, Dr. Sandra Bannock, Dr. James Metcalf, some of the smartest people I know, and all 27 universities around the world that's helped us. Thank you so much for welcoming me here to TEDx in Jackson Hole.